Welcome everyone to our special webinar today. I would like to welcome today's speaker, Isabelle, that is our scientist and service expert. In Immagina, she's mainly responsible for applying our technologies to our client samples, following strict protocols to ensure the quality of our results. Before joining Immagina, she obtained her PhD in biomolecular sciences in Cipio at the University of Trento. She then did her postdoc in the RNA binding proteins, or RBPs, field, aiming to identify and characterize small molecules that inhibit RBPs. Once identified, these molecules hold potential to be used as a therapeutic avenue in cancer and immune diseases. Now, without further ado, Isabelle, the floor is yours.
Good afternoon, everyone, and thanks, Jill, for the introduction, and all of you for joining this webinar that will be on ribosomes as regulatory hubs, their role beyond protein synthesis. Before start digging into it, I just want to remind you who we are. We are Immagina Biotechnology, the ribosome company. Our headquarters is in Trento. In this building, basically, we combine production, services, and innovation. And thanks to our worldwide distributors, we support our scientists and companies to ensure their great research in ribosome and translation control. But now, rather than explain to you who we are, I will go to what we are actually studying. My speech is divided into 4 sections, and I will go through the ribosome heterogeneity, specialization and translation control, ribosome parties, future and technologies. So let's introduce the concept of the ribosome as a regulatory platform. This is a ribosome. It is composed of 80 ribosomal proteins and 4 ribosomal RNAs in most of the eukaryotes, and this divided into different subunits, a large one and a small one, and together they cooperate in driving the protein synthesis. That is one of the mechanisms essential for our life. In fact, in its architecture, the ribosome contains all the sites to enable mRNA decoding, tRNA positioning, and amino acids chain assembly. With this function, the ribosome has always been considered as essential and, for this reason, uniform and passive in its role. But if we look at evolution, we can clearly see that the ribosome is preserving an ancient conserved core, but together with the growing complexity of the organism, the appearance of it has been characterized of the appearance of different structural non-coding sequencing and structures in parallel with the growing complexity of the organism. Therefore, we can consider that the ribosome is a striking example of the concept of the evolution that is characterized by a progressive appearance of non-coding regulatory layers according to the growing complexity of the organism. From the discovery of the ribosomes, already in the 90s, actually, scientists identified variants in the rRNA sequencing and ribosomal proteins, introducing the concept of heterogeneity in ribosome in Xenopus levi. If we go through this timeline, we can see the evidences of this ribosome heterogeneity increased across multiple species, meaning that actually the heterogeneity in ribosome is a hot topic for the evolution. What are specifically these layers of heterogeneity? If we look at the ribosomal structure, several ribosomal paralogs have been identified, and they gave rise to the different organisms. The interactors of the ribosome, the so-called ribosome-associated factors, are responsible for shaping tissues, even the synaptic connection inside the brain. The same concept can be applied for the post-translational modification of this ribosomal protein and their stoichiometry, since they influenced development in several species. And same can be described when we are considering ribosomal RNAs modification in terms of epitranscriptomical one or changes in ribosomal RNA structure. But this doesn't mean that every ribosomal modification results in an evolutionary gain or a distinct phenotype or tissue specialization. In fact, among the many ribosome variants discovered, only a small fraction has been clearly linked to a specific function. So the uncovering of the full range of their role is still ongoing. And in fact, now I will explain to you when a ribosome is heterogeneous but also specialized. If we look at this paper that has been published in 2024, we can clearly see that there is a clustering of the ribosomal proteins according to the different kind of tissues. And indeed, if we are thinking about liver, the hepatocytes, that are the cells inside this tissue, should harbor ribosomes that are really highly active in their translation, since they are to sustain metabolism and secretion activity. Whereas inside the spleen, the immune cells harbor ribosomes that instead are have a more regulated translational machinery, highly dynamic to respond to the diverse external stimuli. But this, again, doesn't mean that there is a population of ribosome for each kind of cell and tissue. In fact, now the recognized model is the one of the different concentration of this population that characterize every cells and tissue type. The situation is more complicated because the cells should be ready to answer to certain stimuli. And instead, in presence of stress, the concentration of the canonical and the specialized ribosome promptly change in order to respond to the stress. But how this specialized ribosome actually affects translation? If we look at the translation process, there are different checkpoints, we can say, that can be regulated by heterogeneous ribosomes, such as the 40S recruitment in the 5' UTR, the start codon selection, the elongation rate, the stopping of the translation, so the reading of the stop codon, and the fidelity. One example of this is given by the iris. Iris are a secondary structure of the messenger RNAs, and in this famous example, this ribosomal protein L38 promptly regulates the translation of the genes by recognizing the iris structure inside their sequences. When there are mutations inside this protein, the neurodevelopment is impaired. Then another regulators of translation are the upstream open-reading frames. These are small sequencing that are inside the 5' UTRs of the messenger RNAs, and they can be bound by different ribosomes and promptly impeding the translation of the main ORF. But what has been observed is that in this case, for example, the ribosomal protein 14 recognize these upstreaming open-reading frame sequences and reinitiates the translation of genes that are regulated by the auxin, that is a plant hormone, and is really pivotal for the proper development of the plant. Actually, these sequences are really important to regulate also stress response, meaning that is a mechanism that is really pivotal. Another layer of translation regulation given by heterogeneous ribosome is due to the interaction with the non-coding RNAs that can be ribosome-associated non-coding RNAs or long non-coding RNAs or SNO RNAs. In the first case, these RNAs directly bind the ribosome in most of the cases inhibiting the translation. Instead, the long non-coding RNAs and the SNO RNAs binds to the ribosome and indirectly mediate interaction with other protein or interaction with the messenger RNA, promoting or inhibiting the translation. The ribosomal associated non-coding RNAs has been described in different species and indeed are an important strategy, for example in bacteria, to respond to external stress such as toxin exposure. Lastly, the ribosome interacts with a lot of factors, the initiation factors, but also other proteins such as RNA binding proteins, protein involved in metabolism, enzyme responsible of post-translational modification, cell cycle regulators, and mRNA modifiers or helicases. And these interaction can lead to the high preference or low preference in the translation of particular mRNAs. One of the most characterized examples is this RNA binding protein, FMRP, that binding this G quadruplex structure inside the RNAs is able to drive this on-off cycle of translation of the specific mRNAs that are pivotal for neurodevelopment. In fact, mutation inside this RNA binding protein leads to the insurgence of pediatric retardation disease, such as the X-linked syndrome. And in fact, there is a cost for this ribosome heterogeneity because these ribosomes are implied in this highly regulated and fine-tuned mechanism. So a single dysfunction inside their function or the mechanism that they are regulated leads to the insurgence of diseases. This is a table in which there is a list of the most common diseases, and what you can see is that at first they are highly tissue specific. So these diseases are affecting bones, brain, and blood mainly. And if we look at the responsible genes for this disease are all ribosomal protein or factors that are implied in the ribosome biogenesis. The ribosome biogenesis is a really complicated process, so it's really prone to error. And these errors give rise to a disassembled dysfunctional ribosome. And for this reason, there is the protein synthesis that is dysregulated and the appearance of the disease. These diseases actually are really rare, but what is increasing is the frequency of the mutation identified for the factors that are regulating this biogenesis, but also the somatic mutation that are of these factors and ribosomal protein that are associated with cancer. Another important thing, in fact, is that, as you can see in this table, this disease, patients affected by this disease, are always susceptible to the insurgence of tumors. And the concept that has been described in order to explain this is this hypo and hyperproliferative paradox. What does this mean? Basically, when there is an individual that is affected by a ribosomopathy, first what happens is that the translational landscape of the ribosome is completely erroneous. This leads to the increasing of the nuclear stress and the activation of the p53 pathway that leads to the death of the cells. The surviving cells are acquiring new mutation, and this clone selection of these cells that harbors these, we can say, oncoribosomes, are then totally transformed in cancer cells. What is the state of the art regarding this kind of pathies. So they are caused by insufficient number of functional ribosomes due to improper biogenesis. They are highly tissue specific, as I was saying. Blood, brain, and bones are usually congenital and related to embryo development. The open question that we have to fill are: why do housekeeping defects affect only certain tissues? How does ribosome heterogeneity or stress signal shape disease outcome? And what mechanism links these ribosomopathies to cancer evolution? Up to now, the therapies that we have are supportive care, so transplantation of stem cells, steroids, and transfusion, and targeted drugs that are entering inside the clinic that are inhibiting actors of the ribosome biogenesis, such as RNase polymerase 1. But what are the future directions? So basically, the gene therapy and the genome editing is entering inside the clinic. The development of safer and more precise drugs for ribosome biogenesis and tagging this stress pathway, and of course, the fact that we have advanced technologies such as genomic sequencing, ribosome profiling, tRNA-seq, and single-cell tool, they are great ways to uncover mechanism and guide future therapies. And in line with this, I would like to show you a case study. We can say that is a pre-case studies. That is this paper that was published this August in 2025 in Nature Cell Biology, in which the authors characterized the mutation of these genes. They also are presenting a molecular mechanism and some therapeutic insight. I just want to disclose that for the lack of time and the purpose of this webinar, I'm not able to show you the entire paper that is full of detailed data and huge amount of models that authors are proposed, but I just present you the major findings. So the study starts with, as I was saying, the identification of this mutation that is related to the insurgence of this severe neurodevelopmental disorder that can be correlated to a ribosomopathy. The study was this mutation has been identified through genomic sequencing and segregation study performed in different families. After characterizing this mutation, the author established an experimental model by editing iPS or reprogramming cells from patient and differentiating them in brain organoids. What they found is that already at day 15 of differentiation, the organoids show a decrease in the size. And when they look on the global translation by performing OPP in vivo labeling and proteomics assays, the global translation is affected by the mutation, such as they observe a decreasing in the abundance of the ribosomal protein. They then want to look for specifically the neuroepithelial differentiation, and what they found is when they performed single-cell RNA-seq, they mapped the results, and as you can see, there is a clear cluster in control condition of the different kind of cells implied in this development process. And what we can already see is that control and mutants organoid are not merging at all, and in fact, at day 15, the distribution of the cell types inside the mutant organoids is skewed and is not following the proper path for the neurodifferentiation. With the immunofluorescence staining, the authors confirmed that actually there is also a reduction in the expression of one of the most characterized marker of the neurodifferentiation, that is BLBP. They tried to assess a molecular mechanism, and they performed single riboseq together combined with RNA-seq in order to study translation efficiency, to understand what is actually translated by this sample. What they found is that there is a group of mRNAs that is downregulated in the mutant condition. And when they went to check which protein were these mRNAs encoding for, they are all protein implied in the ribosome structure, maybe meaning that there are a problem in the ribosome assembly. They went then to look. They then went to look deeply at the structure of these mRNAs, and what we can see is that the transcript that are downregulated harbor this top motif. The top motif is recognized by different stressor pathways mediating translation, such as mTOR. And in fact, if they partially knocked out one of the inhibitor of the mTOR pathway, the area of the organoids is partially rescued, meaning that the pumping of mTOR pathway is re-establishing the normal condition. This is even more true when they went to tagging with a compound that is an activator of mTOR pathway. If you can see that the area of the organoids is rescued. Also, there is a decrease in the apoptosis of the cells, and the expression of the marker of the neurodifferentiation is restored by using this compound. So the model that the author proposed is the following. So there is a normal downregulation of the translation that is physiological and is important for the development. When this downregulation of the translation is dramatic, in the case, for example, of the presence of this variant, the neurodifferentiation is impaired. When this process is somehow restored to a level that is the required translational range, we can go back to a normal situation. You can see that these mechanisms are highly regulated and are really precise and fine-tuned. So in order to decipher them, we definitely need cutting-edge technologies. And as you can see in this table, there is a list of the most common, not all, the technologies that we are now applying that allow us to perform functional and structural studies to analyze the different level of heterogeneity that are inside the ribosome. And Immagina Biotechnology has its portfolio, its own portfolio, all the tools necessary to study heterogeneous ribosome and translational mechanism. I will give you some insight very briefly in this last section regarding these two methods, RAY-APPL, that was developed with our collaborators and scientific advisor in Washington University, and is a method that allows us to obtain cryo-RM structural analysis. And then I will show you the reset that is instead a tool that allow us to perform in vitro translation studies. But before going into that, I just want to give you an example of the application of this ribotRNA-seq, that is basically a combination of our ribolase together with the nanotRNA-seq, that inside one of our internal projects has been pivotal, that we developed in collaboration with Eva Novoa and the group in Barcelona, has been pivotal to study the ribosome heterogeneity in terms of the behavior of the ribosome in choosing different tRNAs in response to different conditions. The workflow is really easy. You can pull down the active ribosome, and then after the small RNA enrichment, you prepare the libraries with the nanotRNA-seq library preparation protocol, and then the libraries are sequenced with Oxford Nanopore Technologies. What we did, we expose the cells to two different stressors, and then we performed RNA-seq, total tRNA-seq with nanotRNA-seq, riboseq, and the ribotRNase-seq. The first stressor that we use is the methionine deprivation at 6 and 16 hours. What we observed is that there is no main change in the tRNA abundance, the total one, and also in the ribo-embedded one. Something, but more significant, start to appear for the ribo-embedded at 16 hours after the treatment. What is dramatically changing is the modification patterns of the tRNAs. We can see, in fact, that inside the total tRNAs, all this blue dot means that there is a decrease that has been confirmed with mass spec analysis of methylation modification in different tRNAs. What we do not observe is the same in the ribotRNAs. In fact, this means that in order to respond to this stress, the ribosome selects the highly modified and proper modified tRNAs in order to promptly respond to this stress, maintaining the steady state level. A different case instead is when the cells are treated with arsenite. The arsenite is one of the most important mediators of the oxidative stress, and in fact we can see that there is a dramatic consequence in the protein synthesis, and in fact the tRNAs that are bound by the ribosomes starts to rearrange in a really different way from the control situation, and this is confirmed also by looking at this volcano plot. And this means that the ribotRNAs are promptly rearranged in order to organize an immediate response to this kind of stress. What we can conclude is that the methionine starvation induces hypomethylation in total tRNA pool, while the ribotRNAs preferentially retain properly modified species, helping to preserve translational fidelity. Arsenite stress, instead, triggers a strong reorganization of the ribotRNAs, revealing a rapid translational rewiring under oxidative stress. So you can see how one of our tools has been really pivotal to study this different behavior of the ribosomes and the tRNAs in different stress.
Now I'm going to present you this method that was published by our collaborators in Nature Communication in 2025, and basically is a purification method of the ribosome using polylysine beads. It is really fast and versatile. You can see that we can obtain ribosome from a low amount of material, in this case cells, and we can visualize with electron microscopy cytoplasmic ribosome, mitochondrial ribosomes, and nuclear ribosome during biogenesis. It is versatile since we can apply this method to different species, as you can see here, and is also functional since it enable to purify ribosomes that are actively translating. You can see here two different strains. The ribosomes of two different strains in bacteria have been purified, and they are able to translate reporter genes. mRNAs. Not only this method is suitable for the preparation and the processing in order to obtain cryo-IM structural analysis with a 2.7 angstrom resolution. So what we can say is that RAPPL rapidly isolates ribosome from minimal material across species. Purified ribosomes remain fully functional, enabling translation and drug testing. RAPPL delivers high-quality samples for structural analysis up to 2.7 angstrom cryo-IM maps. Lastly, this method can be combined with our RESET, reconstituted self-real translation system. Basically, we purified, there are the different elements that are acting during the translation. With the translation buffer, you can detect the translation reaction with then different methods. The application of this methodology vary. You can screen mRNA drugs, test translation inhibitors, and then study the mechanism of cellular stresses. And in this way you can also study the heterogeneous ribosomes. This method was optimized by the one published in Nature Protocols in 2025, but as you can see here, our method, the one that we are developing and will be soon available, is highly performative with respect to the one that was published and respect to the commercial kit. It enabled to see the translation, to check the translation of 2 and more different mRNAs, and is really sensitive to the translation inhibitor drugs, as you can see in here. So with this, I would like to conclude. These are the final remarks and open questions. So specialized ribosome create a fine-tuned translation mechanism. They are hard to dissect, yet crucial for development and disease, and this regulation leads to ribosomopathies and cancer. The technological advances, such as sequencing, proteomics, and cryo-IM, are now opening new windows, and with Immagina platforms and the different technologies that we are proposing, you can study working with low input material and in a high resolution. What are still the open questions that remain to be filled? Is how are specialized ribosome generated and regulated? How can distinct subtypes be systematically identified linked to function? And if these oncoribosome could become therapeutic targets? I hope you will discover it. I want to thank you all for your attention. And if you have any questions, please write them or write us, and we will promptly respond you with the answers. Thank you.